Hi everybody. So today we're going to learn about the cell membrane. I know we touched on it a little bit last week, but I wanted to go over some notes with you. So it is your choice. You might want to take some notes as we go along. However, there is the 3.3 study guide for you to be filling out. So you want to do that after we go through the notes. So you're welcome to take your own notes if you want, but what you need to submit today is the 3.3 study guide. All right? So the cell membrane. Key concept, the cell membrane is a barrier that separates a cell from the external environment. All right? Okay. The cell membranes are composed of two phospholipid layers. So we call this a bilayer. So you can see here, we have our phospholipid head, our fatty acid chain, which is our tail, and then we also have another phospholipid head and a tail facing each other. So on the Jamboard activity, you need to make sure you watch that and correct yours if it's incorrect. And I explain this model. So the phospholipid is made up of, let me get my drawing tool. Oh, very fun. So here's your head, and then these are your tails in here, okay? So phospholipid is made up of a glycerol backbone attached to a phosphate head and two fatty acid tails. So the head, remember, is hydrophilic, meaning water-loving. It's polar. It attaches to the water around it. And the tails are hydrophobic, meaning they're scared of water. They don't like water. Okay? So not that you need to know this, but this is called amphipathic. I can never say that word. I wrote it down so I could try, but I never can. Amphipathic, it's because it's both hydrophobic and hydrophilic, so water-loving and water-hating at the same time. Okay? Oh no. Uh-oh. Okay. Sorry. Okay. So the cell membrane has two major functions. Function number one, it forms a boundary between inside and outside of the cell. So that's function number one, forms a boundary between the inside and the outside of the cell. And number two, controls passage of material. So it decides what comes into the cell and what goes out of the cell, or what doesn't come into the cell and what doesn't go out of the cell. So here's a picture of your cell membrane. So here we have our head. So here's a head, right? Here's our tails, tail and tail. And then we have our heads and our tails. Okay, so this creates that bilayer. So this is the cell interior, cell exterior. So this is the outside of the cell, the inside of the cell, okay? So then we talk about carbohydrates. And if you remember from the reading, a carbohydrate has a specific function, and it's kind of like an identification tag for the cell. It kind of lets the world around it know what that specific cell function is, and it distinguishes it from other cells. Okay, so that's what carbohydrates do. Okay, hydrophobic, water-fearing, hydrophilic, water-loving. So an integral protein and a peripheral protein. So an integral, so inter is inside. So you're inside, so this would be E. Oops, sorry. So your integral proteins cannot be separated from the cell membrane without destroying it. All right, so it's part of the cell membrane. You can't really have an integral protein and you can't take it out without destroying the cell membrane. A peripheral protein is easily removed from the membrane. It's either internal or external side of the membrane. So, sorry, my bad. Your peripheral, wow, that's a horrible E. Your peripheral protein is E. So it can be on either the inside or the outside of the cell. However, if you notice, it's easily removed. So we could like detach it right here. Whereas D is your integral protein. That's a horrible D also. I'll get better, guys, okay? An integral protein that is embedded in the cell membrane, and the only way to remove it is if you destroy the cell membrane, okay? Cell membranes are composed of a 2-phospholipid bilayer. The cell membrane is made of a phospholipid bilayer. There are other molecules embedded in the membrane. Okay, so we talked about carbohydrates, how their identification tags. Cholesterol is another one. So cholesterol is a major part of actually animal cells. So like us humans, we have lots of cholesterol in our cells. It also has a hydrophobic and hydrophilic end in the cholesterol, which kind of makes it similar to the cell membrane, right? In that sense, which allows it to be inserted directly into the bilayer. So you can see these little right here. That's because they share that same property. So your fatty acid chain has a hydrophilic and hydrophobic, so water-loving and water-hating, as well as your cholesterol has that. It kind of changes the fluidity of the cell membrane, all right? So it kind of adds, is it going to be like tight and rigid, or is it going to be really fluid, all right? So that's what the choloesterols do. Proteins we talked about. So we have integral proteins and we have peripheral proteins. So proteins have a very important function. Proteins can be transporters. So they move ions or other molecules from outside of the cell to inside of the cell or vice versa. Proteins are receptors. They allow cells to receive signals from their environment. They can be enzymes, which you guys talked about as catalysts in last unit 1, or they can be just anchors to help shape the cell. So proteins have 4 functions. They can transport material. They can be protein receptors, which accept signals from the environment to trigger action in the cell. They can be enzymes, so catalysts, or they can be anchors to help with cell structure. Okay? We talked about the 2 types of proteins, integral and peripheral. Okay? So we've talked about cholesterol, carbohydrates, and proteins, as well as our fatty acid bilayer, or our phospholipid bilayer. So let's watch this quick video. Ooh, I'm actually going to play it. I'm going to play it up here.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure.
That's that fluid mosaic, right?
The survival of all life.
So you have a fluidity, so it's moving, it's fluid, but the mosaic, meaning you have cholesterols, proteins, fatty acid, phospholipids, you have cholesterol, so it's like kind of like that mosaic look in art where you have like little tiny pieces all put together to make this beautiful picture, right? So I think of like little tiles that make, like, put a bunch of little tiles together and it makes a gorgeous mosaic picture of a fish, maybe. So the same thing here applies. So you have this fluid mosaic model where you have all these tiny little bits and pieces working together and they create this beautiful fluid movement. Okay? All right. Almost done. So cell membranes are composed of 2 phospholipid layers. The cell membrane is selectively permeable. What does selectively permeable mean? So selective permeability means a couple things. The membrane lets some things in, some things out. the membrane lets some things in and some things out under certain conditions. And sometimes the selective permeability lets nothing in and nothing out. So it has the option to choose what comes in and what comes out of the cell at what point of time. So for example, gases, lipids, and hormones are so small that they just kind of squeeze through this little layer and they can get inside and outside of the cell. Whereas bigger molecules, such as macromolecules or ions, we have to use those protein transfers to get in and out of the cell, which I'll get into later. Okay? So here's another picture of selective permeability. So this guy is small enough he can fit right through the phospholipid bilayer. He can go from the outside to the inside. And the membrane says, I like you. You can come on in. So receptors. Receptors bind with ligands and change shape. So let me move this up. Oh no. Can I move me? Oh, good. Okay. So outside of the cell, you have ligand. So ligand comes into the cell and binds with a receptor. So you have two types of receptors that we read about last week. An intercellular receptor is a cellular receptor. There we go. So an intercellular receptor is within the cell. So molecules are transported across the cell membrane, as you can see in this picture, and interact with the cellular receptor inside of the cell. And then that enters into the nucleus, tells the cell to do something. So this ligand enters through the phospholipid bilayer, interacts with the receptor, changes shape, all inside of the cell, communicates to the nucleus, something's going to happen. All right? Something different's changing. Okay? The other type of receptor, so there's two. We just went over the first one, intercellular. We now have membrane receptors. So membrane receptors are a little bit different. Or you can call them molecular receptors. So you can be inside the membrane versus like kind of inside, like literally embedded inside of the membrane. So right here, here's a picture of the receptor. So notice on the last slide, the receptor was inside of the cell, intercellular. This receptor is just located in the membrane. That's why we call it a membrane receptor. Okay? The molecule is too big and cannot cross the membrane like it did in the first one. So it binds with this membrane receptor to change shape and send signals across the membrane to the cell to the nucleus. So the difference, intercellular goes right across the membrane, hooks onto a receptor, sends a signal to the DNA, sends a signal to the nucleus to change something in the gene. And a membrane receptor is located literally inside of the membrane. The ligand binds with this receptor, transmits it across the membrane to the inside of the cell, and sends it away to the DNA. Okay? All right. So that is it for today. You need to complete the 3.3 study guide. Please rewatch this, rewind it. I'm turning my camera on. There we go. Whatever you need to do to understand this. So today is Wednesday. We are going to be doing a lab this week about cell membranes and selective permeability. So hopefully it'll be a little more hands-on and a little more fun for you. Okay? All right. Bye.